Hi you guys. Today we're going to be talking about enriched, selective, and differential media. So the first one we're going to discuss is enriched media. Enriched media contains special nutrients to promote the growth of fastidious organisms. Fastidious organisms are organisms that refuse to grow on general nutrient agar. They're very picky about what they eat, and they can only eat certain things. It's like you're having a really picky kid and he's not going to eat that, but he'll eat this but not that and everything. So fastidious bacteria like Haemophilus influenzae or Neisseria gonorrhea are considered fastidious bacteria because they will only grow on enriched media. So chocolate is the most widely used enriched media. The way I remember it is for chocolate, everybody loves chocolate, so that's how I remember that it's enriched, because chocolate is rich and everybody loves it, so like that, so most all bacteria will grow on it. The next one is a differential media. Now this will allow growth. Okay, so it's letting everybody grow that wants to. Not fastidious bacteria, remember, because that requires enriched. But just your general run-of-the-mill everyday bacteria will grow on this media, differential media. For example, sheep blood agar, but it allows growth while showing different growth characteristics to tell the difference between genus and species. And so, like I said, sheep blood agar is one of these. So this sheep blood agar, if you would take away the sheep blood out of it, it would just be a clear yellow, okay? But when you add blood cells to it, obviously it turns this opaque red. So some bacteria like Staphylococcus aureus like to lyse those red cells, so it's sort of like, say for example you have a bright yellow kiddie pool in your backyard and you fill it up with bright red water balloons, okay? When you look at your kiddie pool from afar, it's going to look like your kiddie pool's red on the inside because of all those balloons, right? But when you start popping all those balloons, when you pop all the balloons and all the balloons go away, then you're going to be able to realize that since all those balloons are gone, you're gonna see that your pool is actually yellow, and that's what happens here. This, say for example, Staph aureus, pop all those red cells around the colonies, right? And now you can see that yellow agar underneath, like your little kiddie pool in the backyard. Okay, so this is called beta hemolysis, and this, because it's a B, beta, and this is a complete clearing of the agar around the colonies, so around the bacterial colonies. One very typical beta hemolyzer is a Staph aureus. Okay, so then if we go here, this is the gamma. Gamma, as you can see, doesn't lyse the cells at all. It makes no change onto the red cells at all. It does nothing. It's just laying on there doing nothing. And this is also called non-hemolytic, so gamma or non-hemolytic. Now the next one here we have is alpha, and this is considered to be a greening or a partial lysing of the red cells. This would be something like a Streptococcus pneumoniae, where it lyses some of the cells, but it doesn't really do that great of a job at it, and so it kind of just partially lyses them, and it changes the color of the agar to this grayish green here. So you can understand why hemolysis, differential hemolysis on the sheep blood agar is so important, because if you can look at it and you see something like this that's alpha, I'm not going to work this up as a strep or a Staph aureus, right? Because I know that Staph aureus is not alpha hemolytic, so I know that it's not Staph aureus. And if I would see something like this that had a huge beta zone around it, and that's one, okay, then I know that it could be Staph aureus, but I know it's not strep pneumo because strep pneumo is alpha. So you can see how that would be important to give you a good starting place about where to go next in your testing if you know the hemolysis of an organism. So the next type is selective media, and this promotes the growth of one type of bacteria but inhibits another type. So one example is a CNA agar. CNA agar stands for colistin and nalidixic acid, and both of these things are antibiotics against gram-negative bacteria. So CNA plate will not allow gram-negative bacteria to grow. So as you can see here on this very low resolution picture, that number 1 is Enterococcus faecalis, which is a gram-positive cocci. Number 2 and number 3 are Enterobacter aerogenes and E. coli, both of which are gram-negative bacilli. So as you can see, those antibiotics, that colistin and nalidixic acid, have inhibited the growth of these gram-negative bacteria. Now here we have Staphylococcus aureus. Okay, you can see the difference in just the colony, just the way they look on the plate, right? But since both of them are gram-positive bacteria, they will grow on DNA. The next one is selective and differential media. This promotes the growth of one while inhibiting the other. That's the differential part, or this, the selective part. But the growth shows different physical characteristics to tell the difference between bacterial types, which is the differential part. One example of selective and differential is MacConkey. Okay, MacConkey is selective for gram-negative bacilli. It contains crystal violet, which is death to gram-positive bacteria, so gram-positive bacteria will not grow. It will also differentiate between lactose and non-lactose fermenters. So what happens is, if it ferments lactose, if it ferments, and you know, fermentation causes acid, which is why when you, some people get heartburn when they drink wine because it's a fermented drink. It creates acid, and as the acid goes up, there's a color change in here. So as you can see, if it's fermenting, it'll turn pink. Okay, but if it's a non-fermenter, it'll stay clear. So it's not fermenting these sugars. So for example, in a stool culture, Salmonella and Shigella are both non-lactose fermenters, but your normal gut flora are all lactose fermenters. So in a stool culture, as you know, if you see something that looks pink, you know, okay, good. I know it's not Salmonella and Shigella. It's probably just normal gut flora. I'm not going I'm done now. But if you would see a non-lactose fermenter, then you would say, all right, well, it could be Salmonella and Shigella, and then you work it up. So that's why that's important too. So this is selective and differential. Differential for the lactose fermentation and selective for gram-negative bacilli. Okay, so what type of bacteria or what type of media promotes growth of one type of bacteria while inhibiting another? Selective, you got it. What type of media contains special nutrients to promote the growth of fastidious organisms? You remember this one? Enriched. Yep, everybody loves chocolate. What type of media allows bacterial growth while showing different growth characteristics? You know what that one is? Differential. Good deal. All right. That's it for this, and I will see you guys in class. Thanks. Take care.